Excellent. So let's gonna go ahead and get started. For those of you who don't usually join us, this is the Center for Genes, Environment, and Health talks. We are trying to kind of bring together the fields of precision medicine, human genetics, and health disparities and populations underrepresented in medicine, both for the sake of improving science for all and improving health in those particular populations. We're really delighted to have Dr. Carlos Bustamante, and I'm going to let Esteban introduce him.
Great. Thank you, guys. It's an honor and privilege to be able to introduce one of my best friends and scientific collaborators, Carlos Bustamante. Carlos and I have collaborated for many years, and it's been an absolute pleasure. So Carlos was originally trained at Harvard. He got his biology degree at Harvard, undergrad and master's in statistics and PhD all at Harvard, and did a postdoc at Oxford. Carlos was a faculty at Cornell up until 2009, and then joined the faculty at Stanford in 2009, and he's currently at Stanford and on leave now. He is the founding director of the Stanford Center for Computational and Evolutionary and Human Genomics, and the inaugural chair of the Department of Biomedical Data Science. He has gone on to do many great things, and one of the most prestigious is he was a MacArthur Genius Award Fellow, and he is now the founder and CEO of Galatea Bio, and he is a venture capital partner at F Prime Capital. So Carlos, welcome, and it's going to be great to have you. Floor is all yours.
Thank you. Thank you, my brother. It's a real privilege and pleasure to be here with you. I'm transmitting live from, as I like to say, my little piece of heaven on earth. This is our little warehouse here in Hialeah, Florida. This is a couple miles from where my brothers and I grew up, especially my brothers. They really grew up here. We moved to Miami when I was in high school, and I do consider Miami my adopted hometown. I'm from Venezuela, and it's hard for us to go home while we're not suffering the same exact kind of conflict that the Ukraine is suffering. We have had sort of 4 million displaced people through just a lot of heartache in my country. So it's great to be able to live in a place like Miami that is sort of on the front lines of many things, including trying to diversify genetics. So and I want to tell you a little bit about what brought me to take on this new challenge. The internet here is sometimes spotty. It's kind of like being in Latin America. So if I pop in and out, I apologize. As Esteban said, I'm still on the faculty at Stanford until they kick me out. We'll see. And I have been able to get some investors to give me financing to lift this new enterprise here. And what we're trying to do is build what we're nominally calling the Biobank of the Americas to scale precision health for all. And so I'll begin by just disclosing my conflicts of interest. I've been privileged to be involved with a series of investments and companies throughout my time. That started at Stanford, and it got so interesting that I sort of left the day job to take this on full time. But most of everything I'm going to tell you about is actually my academic research and a big project that began at Stanford in the early days of the COVID pandemic. And look, everybody knows that we get to be the front man for a pretty cool band, and this is the band that I was able to put together. So Esteban always talks about being an orchestra conductor. He's a classier guy than I am. I'm more sort of a kind of ska band or something. And so I was able to get you and Ashley to come play with me on this one, and Manny Rivas, who's been a dear friend. And we basically commandeered these two NovaSeqs that Stanford had set up to do clinical genome sequencing but they hadn't started when the COVID pandemic hit. So we took them over and we sequenced all the COVID swabs initially with a public health exemption. It was a consent exempt public health exemption, and then we went back and IRB'd everybody to link it to their health record. So I'll tell you that story. And then Alex Ioannidis sort of rolled up his sleeves to help with a bunch of the analysis. And then this really involves also algos that Emer Kenny and Jen Vodchik have helped with. Two really important people. And I want to mention them because of the clinicians don't always get their shout outs. And so Ben Pinsky, who's done like the actual COVID testing and ran that operation, I just like hats off to the guy. Stanford's done 1 million COVID tests. I'm sure you guys have done 1 million too, but it's really impressive what our healthcare systems have been able to prop up. And this is a story of how we were able to kind of repurpose that for research. And then our junior colleague, Dr. Powerek, she sort of took this on as she was standing up her own lab, and it was a sort of separate side thing. She's a cardiologist, but jumped in to help us, and it led into a really interesting project around a passion of mine, which is post-infectious sequela. So I'm going to try to run quickly and tell you how this all maybe hangs together. So what am I trying to do? I'm eventually very passionate about bringing precision health at scale for all, and I want to be able to link underrepresented groups with digitized health records and the ability to recontact, so that I can bring DNA sequencing at scale to those on the tails of the distribution. And the idea is, let's collect a ton of samples and then sequence the ones on the tail. So, we don't have enough money to sequence everybody, but if I collect 10 million samples, maybe I can sequence 1 million of them. And those million are the interesting ones if I can use algorithms to pick up. That's basically the whole idea. So let me take a data first approach, link all the data, and then figure out who to sequence. And as I sequence them, I figure out who might be giving me insights into basic biological processes. And of course, that's not going to just benefit the minority populations, it'll benefit everybody. It's just the right thing to do. And part of the rationale for this is of course what everybody knows now, that human genetics are our best tools for improving both the safety and efficacy of the drug discovery pipelines. And PCSK9s are now the poster child for this. And I don't need to spend a lot of time with this group on this, but just to recall, in my opinion, this is the best example bench to bedside that we have for how we go from a basic finding, namely that the genetics of blood and lipid cholesterol, really translate into therapeutics through monoclonal antibodies, and crank in getting to something you can put in people that lowers cholesterol and averts heart attacks and strokes. So how do you scale this? Well, you like own the UK Biobank, and you have 500,000 samples, and you now do have enough money because you're the UK, so you don't have to do what I do, but that's okay. And you go, strike a deal with pharma, and if you have 500,000 samples with a lot of clinical data, then you might be able to get some really interesting drug targets. And so, and these are just some examples of what the UK has been able to do, and they now have been able to use their resource to get leads for chronic liver disease, multiple autoimmune, neuropsych, cardiovascular. So this is now a proven thing, right? And you just need to read 23andMe's, guidance to investors. I think they've got like 40 drug targets that they're working on. So this is not like a hypothesis. This is like the way drug development is now being done. So if you want to do drug development and include minority populations, like you need to include minority populations. And like, it's just that simple. And so what I want to sort of point out are a couple of things. Number one, this is the now canonical paper with Esteban on genomics for the world. And, a decade ago, we began calling attention to this issue. And the statistic that we drew people's attention to is that 96% of samples were from populations of European descent. And I wish the world had gotten better, but it literally has not gotten any better. Like the statistics have just not moved. And in fact, in some sense, they've gotten worse. So this is an update from Alicia Martin, who was a graduate student with me at Stanford and then a postdoc at the Broad. So she had a very nice paper on polygenic risk score and the translation of polygenic risk scores. So this is the number of GWAS samples as a function of time. This is the fraction of GWAS samples in the catalogs. And so you can just see the explosion of European GWAS. This is the fraction of samples that are of European descent. And if you exclude East Asia, which is really China and Chinese GWAS, then the numbers haven't changed at all. And Hispanics continue to represent, for example, less than 1% of all samples, right? Even though they're about 8% of the world's population. So you're talking about 650 million people that are largely left out of precision medicine. It's about twice the size of the United States. So we view that as problematic, particularly here in Miami. Hialeah, the city I'm in, is the number one foreign-born city in the United States, and Miami's number 2. So literally in my neck of the woods, this is a problem for us. And it's a real problem in that when you try to translate from these polygenic risk scores across ethnic groups, they just don't work, right? You know, this isn't now a theoretical problem. This is a proven out problem, and you just need to read Alicia's paper on this. This is, you know, from one European population to the next, they work pretty well. From a European population to a Hispanic population, sometimes work. It all kind of depends on the trait and the population you're looking at. Across a European and South Asian populations, it's all over the place. You know, Europe to East Asia, not very good. That's why they, you know, basically go and, go do their own GWAS, right? Why else wouldn't China invest $10 billion, right? If they thought this, you could just, you know, pilfer it from the US, they would, right? But no, they have to go, obviously, you know, do this themselves. And then, you know, as per usual, Africa is left out, right? And so of course Africa has to, you know, often take care of Africa. And there's some great efforts now underway here, and there's a, you know, a real need to you know, help organize these efforts. But to me, the data is crystal clear, right? You know, you can't just assume you're going to do everything in Finland and the UK and the world will be okay. Rather, the data is really suggesting if you continue to do what we're doing, we will absolutely widen health disparities. So, you know, what's my vision? Individuals and families need to be able to control their health data, and they need to be able to give others control how it's used, by whom, and for what purposes. And that's why we're, you know, using this biobank in the Americas as a first project, trying to get to 10 million people who can give us access to their health record data, and from their biosamples that we could make use of to potentially get to DNA and then to genetic information. So what's changed that allows us to do this? Again, this is just a reiteration of what I'm trying to do. This is rhetoric, you know, tell them what you're going to tell them what you told them, and tell them again. So I'm trying to build a big biobank and data lake to accelerate precision health in Latin America, focus on underrepresented groups, digitize health records with the ability to contact, and powering the genetics of health and disease. The once in a lifetime opportunity is that the world's now awash in COVID discards, right? And so if you think about, you know, what could have happened had we been able to start from, you know, the beginning, we'd be, you know, much better along, right? You know, if we'd been able to, you know, collect sense from day one, we'd be able to, you know, have already built this, right? There've been, you know, 80 million cases in the United States, and you know, if you would have caught everybody, you know, everybody's already been sampled multiple times. Biden sent 1 billion tests out, but you know, even just floating on the existing infrastructure, there's still plenty to be done. And in many ways, Vanderbilt already proved this out, right? You can get people to give their consent to use the discarded material. And in some sense, I think COVID is fascinating for 2 reasons. One, it's a really great natural experiment from a genetics point of view to open up the genetics of what we think of as comorbidities, but in fact, they're the underlying genetics of traits that led them to be sampled as COVID cases, right? So, and we'll talk a little bit about, who ended up as a COVID case. They were the unlucky, and they were the unlucky for several reasons. They were unlucky either because for socio-demographic reasons, they couldn't get out of the way of COVID. They were unlucky because for genetic reasons, they were on the tail of the genetic risk score, or they were unlucky in combinations because they had, just a bunch of exposures, and this eventually, became insurmountable. There was, of course, a phase of the pandemic that was the kind of uncontrolled phase before you had vaccinations, and then you had the vaccination phase. So all of these provide different windows into how you might be able to stratify the genetic signals. And the chronic phase, of course, is going to be long haul COVID. And that for me is in many ways the calling card, partly because we had been studying a series of post-infectious sequelae in children called PANS-PANDAS that I'll talk about. And that will sort of really form the basis of what I've found just so fascinating, that we've been studying this post-strep in neuropsychiatric disorder that half the world didn't believe was real, and then it turns out that the genetics of that condition overlap with a lot of the genetic signals that we're now seeing in COVID, and so to me it is the Trojan horse that begins to open up both a very genetic angle towards how we get the treatments, and then secondly a very humane angle of, hey, you know what, not everybody responds to infection the same way, right? And we learned it in COVID, but maybe we can take that lesson as a silver lining into other conditions. Not everybody responds to EBV infection the same way. Not everybody responds to, and some of these people end up getting MS. Not everybody responds to strep throat infection the same way, and some of these people end up getting heart disease, and let's remember rheumatic heart disease. Well, maybe it was a strain, but maybe it was also the host genotype, right? And so as a trained evolutionary biologist, to me, these are all fascinating questions, but as a fellow traveler on this planet with you all, these are all questions about, our basic humanity and how we treat each other and how we build, humane healthcare systems. So the basic notion of our enterprise is that, the public is enthusiastic about supplying biodata in exchange for insights about their genome. In a post-COVID world, we can, really marry the ubiquitous testing to an opportunity to store and analyze biosamples if this is done properly with proper consent and buy-in from people. So we really have three legs to the stool. The first is building a biobank of consented samples. The second is building analytics that can mine the data. And the third is to do this in a federated way. And I'll try to quickly sort of walk through each of these. Our real proof in the pudding was this project at Stanford, where we commandeered the 2,000 COVID swabs that were positive in the first part of the pandemic. And again, this is really just an extraordinary effort by my colleagues to sequence the material, get RNA and DNA to do the host and viral characterization, and from there really piece together. And we have two very nice manuscripts that have now been produced on this, and it's just incredible what we were able to learn. And I'll try to walk you through this and its implications for other post-infectious sequelae. So here's the link to the paper, but I kind of really like the schematic. The first is we're able to get host transcriptome data, we're able to get viral genome data, we're able to do HLA typing, and we're able to do low-pass genome sequencing. You can actually do more. You could get a higher coverage genome. This was just really a question of how many samples do you multiplex on a NovoSeq, and I believe this was like the typical same workflow that we were using for the viral sequencing on the set of robotics. So that's how we got to this number, and it's about a 2 to 3x genome. This is really, in some sense, the two more important figures from the point of view of this group and what we were interested in. So these are the typical kind of ancestry figures that many in this group are familiar with. On the top are the COVID positive patients that we saw in the first phase of the pandemic. and the bottom are matched time controls. So, we literally interspersed about 20% COVID negatives. And these are, just to walk everybody through, these are the patients of African American ancestry. These are patients of East Asian ancestry. These are the patients of European ancestry. And I want to focus our attention on these. These are the Hispanic patients of largely indigenous Native American ancestry. And you just see the massive overrepresentation of individuals of high Native American ancestry and Hispanics of high Native American ancestry in the positive COVID cases relative to the negatives. And again, we were seeing this from the genetic data. Obviously, the epidemiological data was also bearing this out. And we were also seeing the protective effects of South Asian ancestry, which had, much to do with two factors. One large one being sociodemographic factors of who can and who can't shelter in place during the first part of the COVID pandemic in the Bay Area. You're able to pull these together in the now typical kind of phylogeny of the COVID strains and look at community transmission and the role of community transmission related to ancestry. Perhaps the most interesting figure is this one, which is sort of self-reported ancestry, self-reported ethnicity over time, genetic ancestry underneath. So the first part, the very first few patients that come into Stanford, of course, look like typical Stanford patients, right? Namely European and East Asian patients. Then there's a series of house staff that gets hit. And then it's basically just Hispanics of high Native American ancestry that are positive at Stanford because everybody else is sheltering in place during the lockdowns of the summer of 2020. So this is, to me, a really, really fascinating picture of what we were living through in that first part of the pandemic. What also became clear, though, is that this wasn't just sociodemographic factors. There were real genetic factors, and HLA, as is often the case, played a role. And here are some of the odds ratios on some of these HLAs, including an HLA-C allele that was highly protective, and it turned out to be a high frequency in individuals of Indian Asian ancestry. And when you actually map this out on the epitope side, it's actually what we hypothesize what Delta broke through, right? So if you remember the first part of the pandemic, there wasn't a lot of COVID in India. And I remember sitting around thinking, this to me looks like old Silk Road patterns of protection, some low levels of protection that could be, some basic amounts of immunity that could come from, old transfers of coronaviruses up and down the old, migratory routes that might be expected to travel with many other kind of things. When you pull this together in an actual mapping point of view, you can add mixture map for signals of severity. So this is, in our paper, and it's one example of how you get at, segments of African ancestry that predispose you to COVID, segments that are, protective, and it's just what you'd expect from, the natural history of this thing, right? You're unleashing a virus the human population has never seen. There's standing genetic variation, and of course, there's going to be, just by happenstance, some genetics that predispose one way or another, right? And so the unlucky are going to get hit, and the lucky are going to be protected through no fault of their own in either way. But you're going to be able to map it, especially pre-vaccination. The other thing that became really interesting is that when you overlay these, they began to hit at the neuropsychiatric sequela that could be coming down the pipe. And we had already been primed and concerned about this. So what I'd like to really do is give you a very speculative foray into where this took us, and in particular because I'm a firm believer that we'll eventually get consilience of data from the mixture of the PheWAS and the GWAS. So from one perspective, we want to be able to take the phenotypic data, see what the health record collision looks like, and then verify with genetics. So if we have a very good way of genetically characterizing patients, even if we're not always able to diagnose them forward, right? We should be able to take for example, what Vanderbilt has done and generate signals of how different individuals with different genetic conditions utilize the healthcare system and then back calculate. So this is the Bustamante kind of method. And she proved this out, say, with cystic fibrosis, right? So a patient with cystic fibrosis is going to use the healthcare system differently than you and I would. So even if in the EHR it doesn't say cystic fibrosis, there should be sufficient data, labs, claims, procedures that you should, given a sample, want to pull that sample and sequence CFTR to confirm whether the person is or is not CF. And maybe it'll be a compound het or it'll be a delta 508 something else, right? So we want to live in that world. We want to begin to build these digital nets that create a dialogue between the utilization data and the genetics data that allow us to build this out. And Stanford, so Vanderbilt has done this with CF, Marfan syndrome, achondroplasia, hemochromatosis, Leigh-Ferré, and really proven it out. I mean, it works beautifully well. That, you know, there's plenty of signal here, and surprisingly, like even something like CF, like 25% of the patients at Vanderbilt that had CF were undiagnosed. You know, it happens, and it happens more often than you might suggest. And then there's some, you know, nice exceptions that prove the rule, like PKU. There are many people who are genetically PKU, but luckily you get your public health advice from the back of a Diet Coke can. Don't give the kid phenylalanine, and that's why you don't phenotypically present with PKU, right? So as you think about this, just from a purely informatics point of view, I can stream claim codes data at you, and eventually you get enough, you know, information that you can almost genotype just from the claims code data, and you confirm by the sequencing. So as you now think about all the data that's accumulating on long haulers or others, you want to use that to subset patients. And we had actually done this in the case of the PANS Panda, which is what I want to bring this towards. We had also done this in the case of FH, because FH for me was incredibly important training wheels. You know, FH is a condition of 1 in 200 people. To me, it's like the poster child of precision health. It's what I started the lecture with on PCSK9s. And yet 90% of patients with FH are undiagnosed, right? You should be able to just knock this out of the park, right? This is high LDL in families. If everybody just got a basic LDL test, you should be able to find most patients with FH in this country. In fact, Nigam and team have built a synthetic map. They've taken all the data. They've been able to commandeer all the data. They've been able to serve this data up through the FH Foundation. And we're still pretty far away from finding all the FH patients, putting them on therapy. Most people who are even prescribed a PCSK9 inhibitor can't get it covered, right? And you know, look, I don't want us to overutilize the resources. Like, I'm perfectly fine. Let's just start them on statins, right? Like, if you look at what do you need to get someone's Repatha covered? It's like, yes, did you try, you know, statins for 4 or 5 weeks and then add ezetimibe? And then if that didn't work, then you're eligible for Repatha. Fine, I'm fine. Like, let's just start putting people on statins that should be on statins because they all have high LDLs. We're not screening enough, right? Like, that's like step one. And this is FH. This is something where, like, there's a causal link between high LDL and heart attacks and strokes. And so if we literally found people with high LDLs early in life, put them on the right therapy, we'd avert heart attacks and strokes in the future. And the frontline therapies are super cheap, right? So there's like this, you know, almost demonstration project that we all want to see happen. And from there kind of go into more complicated conditions. So as I thought about that problem, you know, I've tried to sort of tackle, let's try to do this, you know, almost FH dragnet, right? Let's bring this same idea into Latin America. So we're working, for example, with blood banks in Latin America. Let's screen everybody who's a blood donor. It's like $12 to run, you know, point of care LDL test. We can afford to do that for people who would be willing to consent. And then if you run that and you get people in the family and they're, you know, giving you enough information, then that's, you know, valuable enough to kind of turn the crank on. at the same time, let's take the people who are hugely utilizing the healthcare system because they are, careening through, and some of the people who careen the most are these people who have these post-infectious sequelae. So to tell you about that story, and then the last piece is, it's, you can confirm this genetically. So color, that's now, the big COVID testers started trying to do this, but they were trying to do it forward, right? They're trying to sell everybody a $99 FH test. What you should really do is take all the clinical data you have and subset and test 10% of people. Then it's very straightforward, right? If it's $99 to test people for FH, why wouldn't you, sort of distill it down if you have plenty of evidence that they really are already careening through the system as if they have FH? So what's the tie into long haul and why I think this will work? Well, first of all, there seems to be a fair amount of sequela, as you all know. And it may be as high as 30-40% or as low as 1 in 10. Okay, so even if it's 1 in 10, that's still like maybe 7 million people in the country, right? If 1 million people died of COVID, let's suppose 5 to 10 million people are going to be pretty sick. That's like half as many HIV, AIDS patients in the country. I mean, that's a huge number of people. Like, that's not a small number of people, it's a huge number of people. So it's going to be a huge burden on the system, and it's a collection of conditions, all of which may require different, long-term modulation. So like, we better get ahead of this. And what I'm going to argue is genetics may be predictive in this case as to what kinds of long haul COVID people are getting. And we began worrying about this well before there was this thing called long haul COVID, and partly because we were interested in the space of immunoneuropsychiatry, partly because we had been working with this clinic at Stanford, and I would just want to tell you a little bit about what got me interested in this. So I'll show you the data I was shown. I mean, this is literally like 6-year-old data. So this is my dear friend Jenny Frankovich. She runs this behavioral immune clinic at Stanford. This is her model. This is Jenny's model. So her thesis is you have some amount of immune dysregulation. It could come in different flavors. It could be adaptive immune system, innate immune system, HLA. You have an infection. Could be group A strep, mycoplasma, COVID. Leads to inflammation, disruption of blood-brain barrier, accumulation of antibodies and other cytotoxic cytokines. And this alters the brain, okay? And this could lead to, in our case, the condition we study is this pediatric acute onset neuropsychiatric syndromes. Okay, it's very, pot of conditions, and it's a diagnosis by exclusion, which is an awful kind of world to live in, as you all know. But these kids all kind of hang together in the kind of Anna Karenina way of all of these families are unhappy in their own way. But I think we have this opportunity to really kind of subset them, right? So she would classify these as inflammatory brain conditions that can present with psychiatric symptoms. I mean, it's very kind of top down. She's a rheumatologist by training, and she's built a clinic that has, it's a multi-specialty clinic. And so in her mind, right, she's got primary CNS vasculitis, secondary vasculitis, autoimmune encephalitis, basal ganglion encephalitis. And so she's sort of systematically gone through and tried to classify where these conditions may line up. Okay, so we see all of these kinds of kids that show up in the clinic. Here's the thing that brings them in the clinic in the first place. These kids were very, very highly functional one day, got an infection, and then became highly, highly non-functional. Like, that's the phenotype. Okay, kiddo was perfectly fine, and then after an infection went off the rails. And before COVID, people thought, well, this doesn't happen, it's kind of crazy. Now people are like, no, I know a lot of long haulers. So we were doing this pre-COVID, and it is a really, all I can tell you is a heart-wrenching space. These kids are really suffering. 92% have just really, really debilitating OCD. Half of these kids, are wasting. They have just awful eating restrictions. They all have anxiety, awful mood disorders. I mean, just really, really sick, suffering children who used to be perfectly fine. And they don't respond to SSRIs and other, and other sort of mood stabilizers, right? That's why it was just so, calling us, right? So this is the slide that I saw that Jenny showed me. She showed me this HLA distribution, and she said, "Carlos, do you think this is interesting?" I said, "Yeah, like this doesn't look normal to me. Like this is just like out of the range. This B52 off the charts overrepresentation just does not look representative, right?" And this is what the exact number. It's like a 16-fold odds ratio, and then the B38 is a 5-fold odds ratio, and B55 is a 3-fold odds ratio. Okay, crazy, right? Lots of HLA associations. Well, here's HLA-B54 in Kawasaki's disease. We now believe in Kawasaki's disease, right? We've seen a lot of kids with Kawasaki's. Some of these are pretty big odds ratios too, right? Here's a 7-fold HLA-B15 for Kawasaki's disease. Okay. Now I'm going to show you, this is Kawasaki's disease, or maybe classic Kawasaki's disease. And here is MIS-C, which is multi-inflammatory syndrome in children post-COVID, right? They're similar but different, right? Kawasaki's tends to be little kids. MIS-C tends to be older kids, right? Some overlap in phenotype, not exactly the same, right? But, they kind of begin to smell similarly, right? These are beginning to potentially hang together. So if you had enough cases, you could potentially, separate what's going on in one from the other. This is the case that really got me, riled up. This is June 4th, 2020. This is a previously healthy 14-year-old African-American boy that showed up crazy as can be, strapped to a stretcher in New York City. So this is when it resonated with me. I was like, okay, well, this is going to begin to hit the minority populations because they're the ones that have been overly exposed to COVID. So all of a sudden, PANS Panda is going to go from being this kind of like thing that you see in Stanford clinics from overprotective parents that maybe are concerned about their kids to actually being like just massively impacting minority populations. I just got off a call before this from, Children's Hospital in LA, tons of MIS-C. And I assure you, these MIS-C kids are going to have neuropsych sequelae too, okay? So what have we done? So we had actually built an algorithm that did something like FH. We took all of the medication data for the patients, aligned them, and we were able to subset some of these PANS Panda patients. Sorry, I don't know why this thing just went on autopilot, but basically you're able to take, so these kids take an inordinate number of medications. That's in some sense awful. On the other hand, it gives you a ton of data to subset them. So these kids are on, for example, penicillin, right? Because some of these have had strep infection. They're on NSAIDs, and then some of these end up going on IVIG, which is what you really kind of want to avoid, right? Like, do you or do you not put the kid on IVIG? We were able to demonstrate that there's some kids, this is auto-magically learned from the algo. There's some kids that were very classic, just strep triggered kids that the algo, you didn't put this in the algo just learned this from the medication use. So we had to make a medication alignment algorithm similar to a DNA alignment algorithm. It's all pre-COVID that allowed you to sort of subset the patients just on the medication use. From there, what we're trying to do is take what could be what we're beginning to learn as the genetics of severe COVID and put them together with the genetics of, the coagulation as the potential link between these. So here's a recent review on what we're learning of all the different genetic predictors of COVID relative to endophenotypes and other potential intermediate phenotypes. So genetically, higher red blood cell counts associated with reduced risk of SARS-CoV-2 infection, and kind of crummy white blood cell count presentation, to put it very technically, is associated with worse kind of outcomes, right? So, and in particular, what we think is going to really, pull this together are these polygenic risk scores for different traits, because you put these polygenic risk scores. This is from this is pre-vaccine. So traits for the polygenic risk score predicts who was and didn't show up as at risk of infection. And one of the very first things you see here is that everything related to deep vein thrombosis and the genetics of deep vein thrombosis really was associated with who did and did not show up with an infection early on, right? This is what I'm saying, like, who got COVID, the unlucky. So if you happen to have the genetics of deep vein thrombosis, you were more susceptible to infections and complications from infections that landed you up as a case in early COVID, right? Through no fault of your own, right? So that's what the COVID did. It allowed us to get at that as a signal. The other thing we were able to do is begin to pull together all of the genetics of and polygenic risk scores of CRP and D-dimer and platelets and coagulation, and then relink them back into what we saw with pandas. So I'm going to sort of skip to the punchline and basically show that in our cohort of pandas, where we have this sort of autoimmune condition, the polygenic risk scores of deep vein thrombosis, and here's one for you, Esteban, FEV1, also correlate with age of onset of pandas, right? As well as the genetics of red blood cell count correlate with age of onset at first immunomodulation, right? So in theory, these things shouldn't have one thing to do with the other, but they do, right? So it's early days, but these are the traits that we're seeing. So there's some very real overlap between COVID severity, post-infectious, and what we're seeing in this other triggered disease that we've been collecting a fair amount of data on. So what do we want to do? We're actually going to be building 10-to-1 controls, because I think that's really what we need. We need very, very good controls. The biggest challenge is we don't have enough control data, particularly from these minority populations. So I'm trying to really work with blood bank, create a massive control set from which we can look at deviations and the genetics of these very basic blood tests, because all of what we do in diagnosing, particularly this pandas condition, but also in long haul, will come from basic blood tests. So we need to get at the polygenic risk scores for these basic traits, use those in conjunction to look at and straddle into the severity, and then also cytokines and chemokines, as well as some of the coagulation traits that we've studied. The last thing is, where do I think this goes therapeutically? I think this really becomes, as I like to call the mother of all data plays. If you look at the best-selling drugs, they're all monoclonal antibodies, and they'll soon be polyclonal or combinations of antibodies. And so where this meets is the go-to treatment for pandas, and my guess is it may end up being, unfortunately, COVID. Maybe I'm wrong, maybe I'm not. It is this very, just bizarre world of IVIG treatment, and all of the derivatives of IVIG treatment, and where I think there's just going to be a ton of opportunities tying together this sort of massive data set of individuals from which we've collected DNA, we've collected plasma and serum, We've collected the tails of the distribution of those who are outliers, and potentially, collections of antibodies that could be used to rescue phenotypes, especially as we're trying to study these seemingly rare, but really not that rare post-infectious sequelae. Because my thinking is I've become more and more convinced that more and more chronic disease may be related to continual triggering by infectious agents, including many of the things that Esteban has been interested in asthma, for example. To what degree are the host genetic factors interacting with exposure through these kinds of mechanisms? Okay, last thing that I'll mention is we want to live in a world where algos can travel and not the data, because that's going to be very difficult to do. So today, there's really just two models. One is we centralize all the data, or we share the statistics. That's kind of not modern. So everything that we do sort of lives in this world of federated learning, where we have the ability to have parts of likelihoods and parts of models travel and iterate. So that's really the last thing I wanted to mention. And again, this project was really carried out in conjunction with my colleagues at Stanford, who did a lot of the heavy lift, especially on this early COVID study. I should have really listed here Jenny for all of the joint work on PANS Panda, and of course, Esteban has been a mentor and a friend for a very, very long time. I'm looking forward to working with him in Puerto Rico as we expand out there, because I think we really do need to do more together again. And my engagement is in this post-infectious world, because I think it's the right place to go, and that there were just an inordinate number of interesting problems. So maybe I'll stop there and answer any questions that you may have. Thank you.
Thank you. That was outstanding. I have a bunch of questions. I'm going to just throw one out there, and give the floor to other people as well. I guess the thrombosis part, I think I'd seen that before, and it makes a lot of sense and so on. And thrombosis is something we treat, like hypercoagulability or coagulation disorders. We treat. The medicines have gotten better. They used to be, basically rat poison, but we're coming out of the rat poison age. And so I don't know if you're aware, if anybody else is aware of, are there trials looking at, is this just for infection, or is this for long COVID at all? No,
so it's interesting. Just to your warfarin rat poison analogy, the pharmacogenetics of this is the other piece that's really important. So oftentimes as we try to think about this, the other piece that's left out is, not everybody's going to do as well on one regiment than another. So we have to add that. But I think there are going to be a lot of tools in our arsenal, and it's crazy. Like, the current best treatment for long haul is like apheresis, which is old. Talking about, yeah, I know, right? Like, who's going to vote? Who are the controls?
Not something that's easily scalable, and
and not easily scalable. Yeah, but I mean, the nice thing is that there are also good generics there to try, at least, especially as you're looking in a global setting. But I think we're going to learn a ton, and I think we're going to learn a ton because of the collision course we've unfortunately taken as a society with this.
Other questions?
And the other piece that's fascinating is, we'd forgotten about rheumatic heart disease, and it's coming back in interesting ways. And I know Mark had spent a bunch of time in that part of the world, and in thinking about the role that ancestry had played. It's fascinating that the PGX signals also overlap with where you could and could modulate well.
Great job, Carlos. Always inspiring. Our dear friend Paul Farmer died of hypercholesterolemia of a heart attack. So he was only 62.
Hmm.
Your work is so comprehensive. In one of the slides, were you suggesting that genetic ancestry might be associated with COVID and severity,
Yeah.
particularly Native American ancestry?
Yes. And here's one of the big. Nobody has worked harder than the Native American communities in COVID under control. Highest vaccination rates, higher testing rates. I mean, like, this has been an outstanding effort. Like, hats off. It's been an extraordinary effort.
Well, you know, in our Puerto Rico birth cohort pre-metal, we have about a 5% COVID rate, and we just measured ancestry on the mothers and the cord blood. And we'll have antibody titers. So it's time for us to talk.
Okay, I'm all ears.
Yeah. So exciting. Kiyoki, I see your hand up.
Yeah. Hey, Carlos, that was a great talk. Couple questions with regard to doing these large cohort studies where you're recruiting potentially millions of individuals. What's the quality of the clinical data that you're getting on these individuals? I mean, the phenotyping is so critical. I mean, even with the COVID data that you showed, you know, I wonder about in the indigenous populations, how well, you know, you're accounting for other comorbidities, for example.
Great question. So I'm two thoughts on that. The first is from a clinical trials point of view, what I've come to find out, I didn't expect this, but what I've come to find out is that pharma mostly wants controls. They really want very good controls, and so they tend to trust clinicians in being able to diagnose. You know, so you know the diabetic, sure, I'm happy to believe you've done a good job of finding a good diabetic, and so we're looking to deploy robust, scalable, inexpensive point of care measurements that can integrate as a baseline. So like I'm interested in measuring CRP and LDL consistently. You know, that's actually an interesting problem to me. And then secondly, from a finding patients point of view and diagnosis, my view is we're going to live in an overabundance of data, and it's going to be a signal intelligence problem. And so if I think about the FH example, I have a bunch of data streamed at me. So if I think of it as just there's 100 random claims code, 100 ICD-9 codes that come at you, and you're just classifying them FH, not FH. How many do you need to see before your odds ratio is high enough that you got it right? And my guess is like, it's not even 100. I think it like might be 20, 25, right? So it's not perfect, but you know, like there's lots of examples of very simple kind of little automata that allow you to think about that. And so, and we're actually focusing on claims data, especially, and I learned this from Nigam. Nigam Shah told me, in the United States, anything above $50 gets billed. So there's a very detailed record. It's called claims data. You may not know the value, right? But you know why. And so if you think about lab data, procedure data, diagnostic codes, blah, 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 you know, eventually, right, I don't need the perfect EHR. I just need enough data so that I can classify people in terms of, you know, interest. And ultimately, I want to sequence 10%, right? So it's really, when do I, you know, move them up the Netflix queue of sequencing in terms of the kind of capital allocation.
Yeah, great.
It's a different problem. It's a different problem than a treatment problem.
Right. No, we've been working with claims data and trying to merge that with genomic data for probably 2 decades. And the issue is not within our own system, it's trying to get standardization across systems.
Yeah. But so it's funny. I'm very interested in that problem. I asked Lisa this, and she said, I like claims data because it's easy to send like a claims out. I can email you a claims code algo in the afternoon, right? Like whereas EHR, like that's never going to happen. And one of the things we're trying to do is look at not only across systems but across functions, like in the US versus Andorra, for example. Would you code ICD-9, ICD-10 the same? Because I think the decisions might be different. So like if you're for example, in the UK, let's just think about polygenic risk score for high cholesterol. I actually think you would fundamentally make different decisions. In the United States, if I find out that somebody's on the high end, I'm going to start statins early. In the UK, if I find out somebody's on the low end, I'm going to delay statin use. Right. So those are the two decisions I'm looking. They're the completely opposite. They're the different. I'm interested in different tails, so because I've got different cost functions, and so, ultimately, I think, we'll move from the tails in, but I think if we're focused on the tails, that's where, and if it doesn't work in the tails, then there's no hope in the middle, right? That's the other argument.
Thanks, Carlos.
My pleasure.
Carlos, Kyoki and I were interns together in 1995. He has an awesome data set, and you guys should work together.
Would love to. He got the better hair genes, I have to say.
He's also very, very, probably the smartest in my class.
That's not saying much.
So I have one other thought just about the neuropsychiatric stuff and the immuno kind of neuropsychiatric nexus. So there was this interesting finding a few years ago, and there's probably more on it, and I just don't know the literature that well, but schizophrenia, basically, if you're looking for enrichment of like open areas of open chromatin, it's sort of the usual like neuro brain, blah, blah, blah. But then there's also this huge spike for B cells. And I think this is from like kind of a big GWAS schizophrenia paper. It's basically massive overrepresentation of sort of B cell open chromatin selective sites. And so I guess I'm just wondering if you guys have sort of looked at some of the neuropsychiatric sequelae and thought about kind of whether there's overlap with those, with a subset of, no neuropsychiatric diseases. Yeah.
Yeah. And I mean, the B cells, I mean, it's unbelievably interesting. I think what we've tried to do is, at least from the Panda side, look at flares versus remission and the degree to which you can begin to classify these. Betsy Mellons at Stanford, who's been a really wonderful collaborator on this, and the overlap with rheumatology has been really key here, right? So, for example, like lupus patients, right? Like psychiatry of lupus patients, and when they get because it's like eightfold higher or something like that, right? So I do think there's something there, and the degree to which you can begin to get at the markers that pull them out, I think it's interesting. I mean, I just immunology gets more and more specialized every single day. So, but maybe once you get as single cell RNA-seq data, then maybe that'll be, sufficient to begin to pull it out. But I do think you're right. That's definitely the direction to go.
Okay, well, other questions, or we're over time, so I want to
Okay.
honor the time. And so if no other questions, I just want to thank you again. That was an outstanding talk. It's always
My
just,
pleasure.
really enlightening to have you. So hopefully we'll bring you out again sometime soon.
Anytime. Would love nothing more. Thank you guys.
Take care.
Talk to you soon. Bye.